Good afternoon, friend. In last lecture, I had discussed the what is staining, then what is stain, and the different staining technique. The overview about the different staining techniques. Yes, there are different staining techniques. Yes, for example, simple staining and differential staining. In differential staining, yes, there are acid fast stain and gram staining and endospore staining. Today we will see the next. The staining is a auxiliary technique which is used in microscopy. The stain and the dyes are frequently used in biology and medicines to highlight the structure of biological tissues. Yes, what I am saying, staining is what? It is auxiliary technique which is used in microscopy. The stains and dyes are frequently used in biology and also it is used in medicines to highlight the structure in biological studies. Then the stain, it may be used to define and examine the bulk tissues, then cell population or organelles with individual cells. Yes, stains are used for what? It may be used to define and examine the bulk tissues, then cell population or organelles with individual cells. The bacteria are, yes, keep in mind, the bacteria are microscopic organism. They are mostly colorless, and to visualize them or to study their structure, shape and other structural characteristics, they are mostly colorless. Then, and to visualize them, to study their structures, shapes and other structure characteristics, it requires what? Microscope. As I'm saying, bacteria are microscopic organisms. Then we will see the types of stain. There are basically three types of stain. In that, first, acidic stain, basic and neutral stain. In that first acidic stain, it is negatively charged acid radicals. It imparts color to eosin, acid fuchsin, malachite green and Indian ink. Yes, it is. Keep in mind, it is negatively charged acid radicals imparts the color in eosin, acid fuchsin, malachite green and Indian ink. Next, that is basic stain. Yes, of course, acidic, it is negatively charged. Then what about basic? It is positively charged basic radicals combines with negatively charged particles in cytoplasm and it gives color. Yes, what I am saying, basic dyes or basic stain, it is positively charged basic radicals and it combines with negatively charged particles in cytoplasm and it gives color. For example, hematoxylin, then methylene blue and crystal violet. Then neutral in that both positively and negative neutral stains, it carry both positive charge as well as negative charge and it imparts different color to different components. For example, Giemsa stain and Leishman stain. Yes, neutral stain, it carry both positive as well as negative charge and it imparts the different colors to the different component. Then we will see the different staining techniques. In that, first, positive staining, where the actual cells are themselves colored and appear in a clean background. What is positive? It means the cells are colored and the background, it appears colorless. Then it is the background, it is clear. Then it is positive staining. First, the simple staining, in that a staining in which it provides color contrast but it gives same color to all bacteria and cells. For example, malachite green and crystal violet. Then differential staining, in that we use more than different stain or more than one stain. A stain which imparts the different colors to different bacteria is called as a differential stain. Yes, what is differential staining? A staining or a stain in which imparts the different colors to different bacteria is called as what? It's called as a differential stain or differential staining, which contain more than one stain. Yes, in differential staining it contains more than one stain. For example, gram stain or gram staining, then acid fast stain or acid fast staining, and next that is endospore stain or endospore staining. Then we will see the negative staining, when where the cells remain clear, that is uncolored, and the background is colored, then it is called as negative staining. Yes, are you getting the difference between positive and negative staining? In positive staining, the cells are colored and background is clear or colorless, then it is positive staining. And when it is negative, if the cells are uncolored, that is cells are remain clear or uncolored and the background is colored, to create a contrast to add the better visualization of the image, that is the negative staining. This is the main difference between what, that is positive staining and negative staining. For example, in negative staining, nigrosin and Indian ink. Next we will see the bacterial smear preparation. So what is smear? It is a distribution of bacterial cell on a glass slide for the purpose of viewing them under the microscope or observing them under the microscope. So what is smear? Yes, it is the distribution of bacterial cells on a glass slide for the purpose of viewing or observing them under the only microscope. Then we will see the method of preparation of smear. First, aseptically, a small sample of culture is spread over the glass slide or spread over this slide surface. Aseptically, it means aseptic, to maintain the aseptic condition. If you are preparing the smear, then there is need of maintaining the aseptic condition. It means this, at laboratory level, this procedure is carried out in between two burners. The aseptically, a sample, a small sample of culture is spread over a slide surface. Then then it is allowed to air dry, not artificial drying, it is natural drying. Then next step, it is heat fixation to help the cell adhere to the surface. What is the role of heat fixation, or what is the need? As I said, I have already discussed in last lecture what is heat fixation and what is the need of heat fixation. The heat fixation helps to cell adhere to the surface. And then the smear is now ready for staining. Then smearing techniques. First, heat fixation, and second, methanol fixation. First, in that heat fixation, pass air dry smear through a flame for 2 to 3 times and do not overheat. Heat fixation. Pass the air dry smear through the flame 2 to 2 or 3 times and do not air overheat or over dry, and allow the slide to cool before staining. Then methanol fixation. Place air dry smear in a Copeland jar with methanol for 1 minute. Alternatively, float the smear with methanol for 1 minute. Then drain the slides and allow to dry before staining. These are the smearing techniques. First, heat fixation, and second, what it is, methanol fixation. Then we will see the simple staining. Also, it is discussed in last lecture in detail. What is need? Sorry, I have seen the introduction, definition, then procedure, mechanism and application of simple staining. First, Loeffler's methylene blue. It is generally the most useful. It shows the characteristics, morphology of polymorph, then lymphocytes and other cells more clearly than do stronger stains such as grain stain and dilute carbolfuchsin. Then second, polychrome methylene blue. This is made by allowing the Loeffler methylene blue to ripen slowly. The slow oxidation of methylene blue, it forms a violet compound that gives the stain its polychrome properties. The ripening, it takes 12 months or more to complete, or it may be ripened quickly by the addition of 1% of potassium. Then, in contrast to the blue staining of most structures by methylene blue, the violet component stains acidic cell structure. It is red purple. Then dilute carbolfuchsin. It is made by diluting the Ziel-Nielsen stain with 10 to 20 times its volume of water. It is prepared or it is made by diluting the Ziel-Nielsen stain with 10 to 20 times its volume of water. Stain for 10 to 25 seconds and wash well with water. Then overstaining, it must be avoided as this is an intense stain and prolonged application color the cells protoplasm in addition to nuclei and bacteria. Then we will see the requirements: Loeffler methylene blue, then dilute carbolfuchsin, distilled water, compound microscope, cedar wood oil and fixed smear. These are the requirements for simple staining. First, Loeffler methylene blue, then dilute carbolfuchsin, then distilled water, then compound microscope, then cedar wood oil and fixed smear. Then we'll see the procedure. Make a thin smear on a glass slide, then heat fix the smear by passing the slide 2 to 3 times gently over the flame with a smear slider. Then pour Loeffler methylene blue over the smear and allow to stand for 3 minutes. Then wash the stain smear with water and air dry it. Then observe the smear first under low power objective, that is 10x, and then observe under oil immersion objective, it is 100x. Keep in mind the 100x is called as oil immersion objective. And observe the slide presence of organism and also the cellular contents of sample. Here we can see the image of simple stain. Yes, we can see the image. Then we will see the gram staining. This is all about what? Simple staining. Next we will see the gram staining. The gram staining is most widely used differential staining in microbiology. It is the most widely used differential staining in microbiology. It differentiates the bacteria in two groups, that is gram positive bacteria and gram negative bacteria. It differentiates the bacteria in two groups. First, gram positive bacteria and second, gram negative bacteria. First, gram positive bacteria. These are the bacteria or they have a thick cell wall of peptidoglycan and other polymers. Yes, gram positive bacteria, they having the thick cell wall of peptidoglycan and other polymers. And peptidoglycan, it consists of interweaving the filament and which is made up of alternating N-acetylmuramic acid and N-acetyl glucosamine monomers. Yes, peptidoglycan, it consists of interweaving the filament which is made up of which is made up of alternating the N-acetylmuramic acid and N-acetyl glucosamine monomers. Next, that is gram-negative bacteria. They having the outer membrane of phospholipid and bacterial lipopolysaccharide outside of their thin peptidoglycan layer. The outer membrane, it protects the gram-negative bacteria against the penicillin and lysozymes. Yes, gram-negative bacteria, they have a outer membrane of phospholipid and bacterial lipopolysaccharides outside of their thin peptidoglycan layer. And this outer membrane, it protects the gram-negative bacteria against what? Penicillin and lysozymes. Then we will see the procedure for gram staining. It consists of basically 4 steps. First, primary staining, then mordanting, then decolorization, and last counter staining. In that first primary staining, the smear is covered with crystal violet for 1 minute and washed with water. The smear is covered with crystal violet for 1 minute and washed with water. Then mordanting, it is then covered with grams iodine and kept for 1 minute and washed with water. Then decolorization, the smear is covered with alcohol and it is washed with water immediately. Then counter staining, the smear is then covered with safranin and kept for 30 seconds and it is washed with water. And last, this slide observed under microscope. I am repeating the steps. First, primary staining. In that, what is? The smear is covered with crystal violet for 1 minute and it is washed with water. Then in continuation, second mordanting, it is then covered with grams iodine and kept for 1 minute and washed with water. Then in continuation, the decoloration, it is the smear is covered with alcohol and is washed with water immediately. Then in continuation, counter staining, the smear is then covered with safranin. The smear is then covered with safranin and kept for 30 seconds and washed with water. And last, it observed under microscope. Here we can see the structure of gram-positive bacteria and gram-negative bacteria. And what is the result? The bacteria that manage to keep original purple dye have only got a cell wall. They are called as gram-positive. Bacteria that manage to keep the original purple dye have only got a cell wall. They are called as gram-positive. And second, gram-negative, the bacteria that lose the original purple color and can therefore take up a second red dye have got both cell wall and cell membrane. They are called as gram-negative. Yes, what is result or what is observation? First, the bacteria that can manage to keep the original purple dye have only got a cell wall. They are called as gram-positive. And the bacteria that lose the original purple dye and can therefore take up a second red dye have got both cell wall and cell membrane, then it is called as gram negative. Yes, then it is called as what? Gram negative. Yes, gram staining, name after Hans Christian Grams, differentiate the between gram positive purple and gram negative pink stain, and it is used to identify the certain pathogen. It is scientist, that is the name of scientist Hans Christian Grams. It differentiate between the gram positive, it is purple, and gram negative, it is pink stain, and it is used to identify the certain pathogen. Then we will see the principle of gram staining. The gram staining is used to determine the gram status to classify the bacteria broadly. And second, it is based on the composition of their cell wall. Next, the gram staining, it uses the crystal violet to stain the cell wall. Then iodine is used as a mordant, and fission or saponin used as a counter stain. Yes, what is the principle? The gram staining is used to determine, in principle, first thing, that is the gram staining is used to determine the gram status. This point are also in practical, the gram staining, then acid fast staining, negative staining, positive staining, then industrial staining. All those things are in practical. Yes, in principle, first the gram staining is used to determine the gram status to classify the bacteria broadly. Then it is based on the composition of their cell wall. Then the gram staining, it uses the crystal violet to stain the cell walls. Then iodine is used as a mordant. Then fuchsin or saponin is used as a counter stain to mark the all bacteria. The gram status is important in medicine. The presence or absence of cell wall will change the bacterium susceptibility to some antibiotic. Yes, keep in mind, the gram status is important in medicine. For what is important in medicine? The presence or absence of cell wall will change the bacterium susceptibility to some antibiotics. The gram positive bacteria stain dark blue or violet. Their cell wall is typically rich with peptidoglycan and lacks the secondary membrane, and the lipopolysaccharide layer is found in gram negative bacteria. Yes, keep in mind what I am saying. The gram positive bacteria stain the dark blue or violet. Their cell wall is typically rich with peptidoglycan and lacks the secondary membrane, and the lipopolysaccharide layer found in gram negative bacteria. Then we will see the steps in gram staining. As I discussed the steps, crystal violet, it acts as a primary stain. The crystal violet may also used as a simple stain because it dyes the cell wall of any bacteria. Then grams iodine, it acts as a mordant. It helps to fix the primary dye to the cell wall. Yes, it acts as a grams iodine, it acts as a mordant because it helps to fix the primary dye to the cell wall. Next, decolorizer, it is used to remove the primary stain, that is crystal violet, from gram negative bacteria, that is those with lipopolysaccharide embedded in their cell wall, that is LPS. And decolorizer is composed of what? It is composed of organic solvent such as acetone or ethanol, or it is the combination of acetone and ethanol. Yes, what I am saying, the decolorizers is composed of what? It is composed of organic solvents, for example acetone or alcohol, or sometimes it is combination of both, that is the combination of acetone and ethanol. Then finally, last, the counter stain, that is saffronin, is applied to stain those cells, that is gram negative, that having lost the primary stain as a result of decoloration. Then here we can see the images or difference between the gram positive and gram negative. To distinguish the different kinds of bacteria into separate groups based on staining properties, that is two types, gram stain and acid fast stain. Here we can see the gram positive. The red color, it is cell membrane, then black it is peptidoglycan, and then green it is outer membrane. Here we can see the difference between gram positive and gram negative bacteria. Then we will see the differential stain of gram stain. First, crystal violet, that is primary stain. The color of gram positive cell, it is purple, and the color of gram negative, it is purple. Then modern, it is iodine. The color of gram positive cell, it is purple. Then second, the gram negative cell, the color of gram negative cell, it is also purple. Then third step, that is decolorizing agent. In that, what we use, that is alcohol and acetone. The color of gram positive cell, keep in mind, the color of gram positive cell, it is purple, and the color of gram negative cell, it is colorless. Keep in mind difference. And last, the counter stain saffronin. The For that, the color of gram positive cell, it is purple, and the color of gram negative cell, it is red. Then this is the procedure for gram staining. Methyl violet primary stain, that is main stain. Then iodine, then alcohol and acetone, and last carbol fuchsin, that is counter stain. Here we can see the structure and reactivity of gram staining. The heat fixation, then crystal violet, iodine treatment, decolorization, and last, that is counter stain. Here we can see the difference when it is purple and when it is pink, and when it is purple and when it is colorless. Here we can see for decolorization, in gram positive it is purple, and in gram negative it is colorless. And last, for counter stain, that is fuchsin. In gram positive it is purple color, and in gram negative it is pink color. Here we can see the in detail structure of gram positive bacteria and gram negative bacteria, along with each and every part. Then here we can see the difference between gram positive and gram negative bacteria. The cytoplasm, which is present in both gram positive as well as gram negative bacteria. Then cell membrane, it is present in both gram positive as well as gram negative bacteria. And then peptidoglycan, it is also present in both gram positive as well as gram negative bacteria. And last, that is sorry, the lipopolysaccharide, which is only present in gram negative bacteria. Here we can see the images for gram positive cocci and gram negative bacilli. I will stop here. The remaining part we will discuss in next lecture.